Hello students, myself Muhammad Sharif, Assistant Professor, Department of Biochemistry, Islamia College, Vanambadi. Today we are going to discuss about structure of insulin receptors. First of all, we know what is insulin receptor. Insulin receptor is a transmembrane receptor that is activated by insulin. IGF-1 and IGF-2 belongs to the large class of tyrosine kinase receptors. So here we used to discuss about the insulin receptors action and how it works, that we will discuss here. First we will see the introduction. Insulin is a peptide hormone produced by beta cells of the pancreas and responsible to nutritional stimuli. Main sites of action of insulin in the body include liver, adipose tissue, muscles and brain. Insulin is secreted by beta cells in response to an increase in blood glucose level. And also insulin binds to the insulin receptor and facilitates uptake of glucose by the cell. Insulin's interaction with the cell triggers both metabolic and mitogenic cellular responses. Insulin, like growth factor IGF-1, is a mediator of cell growth and differentiation. Insulin receptor belongs to a family of receptor tyrosine kinase which phosphorylate their substrate proteins on tyrosine residues. The insulin receptor comprises of two subunits, that is the extracellular alpha subunit and the transmembrane beta subunit. The functional receptor exists as a dimer, that is heterotetrameric complexes alpha 2 and beta 2. So the alpha subunit contains the ligand binding site, as it is the only subunit identified by affinity, that is labeling protocols, and then the beta subunit is involved in intracellular signaling. The main site of action of insulin is liver as well as muscle and adipose tissue. So here we can see how insulin works, that means glucose is how works through insulin, that we can see here. A clear chart is given here. You can see how glucose is converted to fatty acid, then fatty acid to triglyceride, that means adipose tissue, from adipose tissue to fatty acids. Again that fatty acid goes to protein, that protein available in our muscle, from that amino acid or glycogen in liver, that finally converted to glucose, mainly stimulated by insulin, increased by feeding, inhibited by insulin, increased by fasting, and in diabetes condition. In this chart you can see how insulin is distributed. Then here also you can see that how insulin spread through three major parts, that means adipose tissue, liver and striated muscle. In adipose tissue, glucose uptake will increase, lipogenesis will increase and lipolysis will be in decreased condition. In liver, glucogenesis downs and glycogen synthesis increase and lipogenesis also increase. In striated muscle, glucose uptake also high, glycogen synthesis will be high, then protein synthesis also will be high. So here we can see the insulin action. It stimulates glycogenesis, lipogenesis and protein synthesis, and it inhibits mainly glycogenolysis, lipolysis, protein breakdowns. Then how insulin action in liver, that means it decreases the glucose output, decreased gluconeogenesis and increased glycogen synthesis, and also decreases ketogenesis. Muscle cells and adipose cells in insulin action. Markedly increased uptake of glucose, and present on virtually all cells, high concentration in liver, muscle and adipose tissue. Insulin receptor belongs to tyrosine kinase class, already we saw in introduction. Here we can see the action of insulin receptors from alpha and beta, and GLUT4 also you can see here, MAP kinase signaling pathway, PI3K signaling pathway, and further MAP kinase to cell growth, proliferation and gene expression. In PI3K signaling pathway, the synthesis of lipids, proteins, glycogen and cell survival and proliferation. In GLUT4, glucose transporter is stimulated by insulin in muscle and adipose tissue. Half-life of insulin. Insulin receptors has 7 to 12 hours and insulin in 5 minutes. GLUT4, two major insulin target tissues are there. One is skeletal muscle, another one is adipose tissue. In GLUT4, sequestered mainly within an intracellular compartment, it does not function as a glucose transporter until insulin signaling causes translocation of GLUT4 to the cell membrane. Glucose disposal after a meal. Important sites of glucose disposal is skeletal muscle and adipose tissue. Important sites of glucose disposal is and also skeletal muscle and adipose tissue. Insulin is responsive for that. Exercise increase insulin sensitivity of muscle. Persist for several hours after exercise, that means regular exercise, it prolonged increase in insulin sensitivity and decrease in insulin receptors per cell. It exposure to increased level of insulin in diabetes 2, obesity, acromegaly. Normal glucose range is 70 mg per deciliter. It stimulates insulin synthesis and insulin secretion. Food intake will be insulin secretion stimulated. Insulin is a anabolic. Insulin helps in the storage of carbohydrate, fats and proteins. Insulin promotes energy storage, conversion of substrates into glycogen, triglycerides and proteins. Excess carbohydrates stored as glycogen in liver and muscles. Excess carbohydrates that cannot be stored as glycogen converted into fats stored in adipose tissue. What is insulin receptor? That means when insulin interacts with the receptor, dimerization of the receptor takes place. Due to this, the cytoplasmic domains of the receptor come close together, facilitating autophosphorylation and triggering the signal transduction pathway. What is the major role of insulin receptors? It is metabolically, the insulin receptor plays a key role in the regulation of glucose homeostasis, a functional process that under degenerate conditions may result in a range of clinical manifestations including diabetes and cancer. Insulin signaling control access to blood glucose in body cells. Here you can see the receptors working area, that means in cytoplasm how it binding of insulin to receptor proteins. In next one, phosphorylation of receptor, tyrosine kinase now started to activate. Then in further step, phosphorylation of signal molecules convert to cascade of effect, then glucose uptake and anabolic reactions are take place here. Then we will see the types of signaling mechanisms. Insulin's interaction with its cell surface receptors triggers both metabolic and mitogenic cellular responses. In order to this, insulin employs two kinds of pathways, RAS dependent and RAS independent. RAS is a member of large family of small molecular white GTP binding proteins. Many RTKs are known to activate the RAS protein in order to mediate signals. RAS dependent pathway. Upon insulin binding, the RAS protein is phosphorylated, leading to activation of mitogen activated protein kinase, that is MAPK pathway, which is involved in cellular growth and proliferation. Then RAS independent pathway, in this case another pathway known as the PI3-AKT signaling pathway, is activated by a series of events facilitating glucose uptake by the cell. Types of signaling mechanism: how it takes survival signals, you can see insulin receptors, proliferation, vasoconstriction, GLUT4 translocation. Also you can see here clearly glycogen synthesis, protein synthesis, and autophagy. RAS independent pathway, that is PI3-AKT signaling, proteins involved in PI3-AKT pathway, you can see here. Then next one is the role of proteins involved in PI3-AKT signaling is insulin and insulin receptor. When blood glucose level rise, insulin released by the beta cells of the pancreas binds to the insulin receptor on muscle cells and or adipocytes and facilitated dimerization of the receptors. As a result of this dimerization, the receptor is autophosphorylated at specific residues. Insulin receptor substrate is a family of proteins that are primarily activated as a result of autophosphorylation. The amino acid residues of these proteins are known to be phosphorylated. There are two types of insulin receptor substrate, that is IRS1 and IRS2. IRS1, that is insulin receptor substrate 1, is known to function in glucose metabolism, while insulin receptor substrate 2 functions in lipid metabolism. However, both of these are required by the insulin receptor as well as IGFR to exert different kinds of effects. PI3 kinase, that is phosphatidylinositol kinase, is activated by IRS1 and act as adapter protein. Activated PI3K serve to phosphorylate phosphatidylinositol on the D3 position of these inositol ring generating proteins in 3, 4, 5 and P3. These specialized lipids require pH domain containing proteins such as AKT to the plasma membrane. Protein kinase B or AKT helps in translocation of GLUT4 storage vesicles to the plasma membrane by a series of events leading to activation of RAB proteins. As GLUT4 vesicles fuse with the plasma membrane, number of receptors increase, thereby increasing glucose uptake. Here RAS dependent pathway, you can see general representation of MAP kinase signaling. The pathway: insulin binds to its receptor and causes dimerization. This event facilitates autophosphorylation at the specific tyrosine residues within the cytoplasmic domain of beta subunit of the receptors. The phosphorylated tyrosine residues are docking sites for a number of proteins. In this case, primarily two kinds of proteins are activated: insulin receptor substrate IRS and SHC family of proteins. Both these proteins contain the PTB domain that interacts with the phosphorylated residues on the activated receptor. In case of signaling via insulin, both IRS and SHC are important as indicated by these experiments. This pathway interference with PTB domains of both IRS and SHC blocks the ability of insulin to elicit cellular responses along both the branches of insulin signaling. IRS proteins contains pH domain that facilitates interaction with specific plasma membrane sites adjacent to the receptors. In this unit, our last topic will be internalization of receptors. That means a receptor-mediated endocytosis process that results in the movement of receptors from the plasma membrane to the inside of the cell. The process begins when cell surface receptors are mononucleated following ligand-induced activation. That means ligand internalization is a receptor-mediated endocytotic process in which the cell will only take in an extracellular molecule if it binds to its specific receptor protein on the cell surface. It is an important cell signaling event, and we offer a variety of fluorescent ligands targeted to common receptors. Thank you, my dear students.